Good morning, everyone, and welcome to the April HDSA research webinar. I am very pleased to be here with Dr. Jack Riedling, who's going to talk about neural stem cells as a therapeutic candidate for Huntington's disease. But I wanted to first address, for anyone who's listening in, there was a recent press release that came out from Ionis Pharmaceuticals about their Huntington-lowering drug trial, and they're continuing to release bits of data. They announced yesterday that in the first phase of the trial, lower levels of Huntington corresponded with better scores on clinical tests. And we've had a lot of questions about this, so I just wanted to take the opportunity to remind you that though this is another piece of very promising data. We're not yet able to say whether the drug is going to help people's symptoms. It's still only been tested in a very small group of people, and a much bigger trial will be needed moving forward. It's a very tightly regulated process to make sure that a drug is safe and effective. So there hasn't really been any change in the info or any announcement about details, but we're hoping to share an FAQ and a statement a little bit later today on our website and social media, a little more about that announcement. So thanks for joining in today. And I just want to remind you, I want to remind you how to ask questions during this session of our speaker. So you can go into the questions panel on the right side of your screen and enter a question at any time during the talk, and then at the end of the session, we're going to be fielding those questions for Dr. Riedling. And I also want to remind you that to view this webinar again or to pass it along to anybody who wasn't able to join us today, within about a week, you'll be able to access it on the HDSA website and also on our YouTube channel. So you can go to hdsa.org/research-webinar or connect to our YouTube channel at the top of the screen that's circled there. And I just wanted to mention that our next webinar is going to be on Thursday, May 24th from 12 to 1 EDT, and this will be a presentation about exercise and research into its benefits in Huntington's disease by Lori Quinn, who's a physical therapist and professor at Columbia University. So without further ado, I'm very happy to be able to introduce Dr. Jack Riedling, who is a project scientist at the Institute for Memory Impairments and Neurological Disorders at the University of California, Irvine. And he told me that he and Dr. Leslie Thompson were in grad school at the same time, and that he has a distinct memory of when the HD gene was identified in '93. A lot of his work has been in cell molecular physiology and molecular biology, and in 2009, he and Leslie reconnected to collaborate on studies of neurodegenerative disorders, which led to his current position, where he provides technical and intellectual support for Leslie's research lab and the work that's specifically funded by the California Institute for Regenerative Medicine. Dr. Riedling is a multidisciplinary researcher who's investigated many aspects of small molecule transport and regulation. He's received National Research Service Awards and Research Career Development Award from the NIH, and has created mouse models and looked at disease processes in the liver, intestine, and brain. And today he's going to talk about his research in mice on how neural stem cells might be a potential way to treat Huntington's disease. And so now I'm going to hand it over to Dr. Riedling.
Hi, everybody. Thanks for attending, and I'm really pleased to be here to tell you about our work. I wanted to start off with giving a little more details, thanks for that introduction, about my path to becoming an HD researcher. I think a little bit of personal information will kind of put my position in context. So as was mentioned, I was a graduate student at the same time as Leslie back in the 90s here at UCI, and it was really exciting to be down the hall from them when they mapped the gene and found the Huntington gene. And I followed the work surrounding Huntington's disease peripherally, but my path was completely different, and I was more of a nuts and bolts investigator looking at molecular biology and biochemistry, and my path took me through multiple model organisms, including E. coli. I worked in jellyfish, and I worked in Neurospora crassa up at UC Santa Cruz for a couple years as a postdoc, working on the proton pump, and again, just kind of molecular mechanisms. And then my path took me back to UCI, working on nutrient transport systems in the digestive system. And while doing that, I had developed a couple mouse models, and we had this mouse that had some impairment in its neurology, and I didn't understand it. I wasn't a neuroscientist by any means, or didn't know anything about the brain. So I contacted Leslie because she was still available to me, and I said, hey, what do I do with this? Do you have any advice? And she said, you know, it sounds like you really need to talk to Frank LaFerla, and he's well known in the Alzheimer's field. And so she connected me with Frank, and that turned out to be really opportunistic for me. And I ended up moving my research from the lab I was in to Frank's lab, and collaborating with him to develop this understanding of nutrient deprivation and loss of nutrient transport in an aging brain, and how that impacts Alzheimer's disease. And kind of sadly, on a personal note, around that same time, my mom was diagnosed with Alzheimer's. So that was the first time in my career where I really had a personal connection to my research, potentially helping a family member. And so I applied for multiple grants and working with Frank. And I think if you know of the National Institute of Health, you have to have a really tight proposal to get funding. And my funding proposals were always scored high, but I think they didn't really understand my position, and I didn't have an independent professor track position. So I think that might have been the challenge for me getting grants. But fortuitously for me, at the same time, Leslie's lab, who was again right next door to Frank's, had a need for this CERN project and some mouse expertise. So we didn't know how this was all going to play out, but she had a need and I needed a job. So we started working together, and that collaboration has just been unbelievable and has led me to working with graduate students and helping to expand her lab. Since I've been with her, we went from 7 people, now we have 25, and I get to participate in all kinds of the research projects going on in the lab, mainly driving this CERN project. And I think many of you know Leslie, and she's just one of the most awesome people to be around and work with. And again, I now have a new connection to my research and my work, because we have patients and patient advocates coming to the lab and telling us how proud they are that we're striving to help their families. And we work with people like Frances Saldana, and her story is so touching. And my work now has become much more personal and kind of guided by this incentive to do something positive for families. And so I hope I can tell you a little bit about this story with our neural stem cells, and convince you that there is promise for this type of work. So let's see. So I'd like to just start off with just an outline of telling you what I'm going to talk about today. I have to give some background. I know the HD community is quite knowledgeable on the science, and I'm not going to assume that you know everything, but I'm going to challenge you with some slides that are kind of scientific, but hopefully I can explain them to you. So please focus on what I'm trying to say and don't get caught up in the busyness of some of my slides. So I'll kind of set the table with some background information of where I think needs to be highlighted in Huntington's disease. And I do need to tell you a little bit about what stem cells are, and kind of where they might be useful for neurological disease research and therapies. And then I'll go into specifics about our candidate for this project. So here's kind of rundown of the facts that many of you probably are well aware of. Huntington's disease is considered a rare disease, but the estimates are probably underrepresented because of the nature of defining the disease. And as you know, there's this midlife onset. It's autosomal dominant with a 50% chance of passing down the disease gene to the offspring, to children. It's a very complicated disease that progresses with chorea being aggravated and increasing by degrees over time. And I think everybody knows these initial symptoms of personality changes, movements in the chorea, and the dementia with speech and irregular movements. And you know that this was probably that this was defined by George Huntington in the 1800s. I really want to point this out, this motor impairment and the some of the cognition impairment, because that's things that we look at in our mouse models. So kind of keep those in mind, and I'll discuss our results in the mice in a few slides. So we consider Huntington's to be a genetically programmed neurodegenerative disease. And what I'm showing here is the changes that occur in the brain post-mortem with a control brain on this side and the Huntington's disease a brain on this side. And I think it's obvious that there's massive cell loss in specific areas of the brain. And what I want you to focus on really is the striatum, this area here where you can see this severe degeneration and atrophy of a very specific type of neuron called medium spiny neurons. And what I'm showing over here on the right side is cells grown in culture. And what I want to point out is this medium spiny neuron with this small orange dot that is kind of the hallmark of Huntington's disease, these nuclear inclusions that occur as the disease progresses. So keep these in mind. I'm going to come back to this idea of a nuclear inclusion when I'm pointing out some of our data. Now, as we mentioned earlier, you know the Huntington's disease gene is known, it's been mapped, and there was huge excitement back in the 90s that finding the gene was going to lead to therapeutics, and this is kind of the overall theme that happened when in the 90s when we were looking for genes, that the anticipation of finding therapeutics was going to be much easier. It has happened for certain genes, but Huntington's is very complicated, and so as you know, we're still struggling with trying to figure out what even the normal function of the Huntington's gene is. But let me remind you that the disease is caused by an expansion in the gene of this CAG triplet codon, and once you get over 36 repeats, you progress into the disease state, and the correlation of the expansion with disease onset is well documented. And then this expansion is a poly-Q disease in the protein, and this, what I want to point out here is that this protein is huge. I've worked with a lot of other proteins and trying to understand their function, and Huntington's is gigantic, and it has a lot of areas of regulation, and people are trying to understand exactly what's going on with the normal function of the protein and how it impacts multiple areas in a cell. And so the normal function isn't really known yet, but I'm going to advocate for its potential as a scaffold for selective autophagy. And one of the reasons for this is that I think it makes a lot of sense, but the other is because this is work coming from our lab, and I of course am a little biased. And this is mainly work by Joan Steffen with a former graduate student, Joseph Ochaba. And so what is autophagy? So autophagy is really self-eating, if you will. And so it's the way that our cells kind of clean house. They get rid of damaged proteins and kind of recycle them or take out the trash. So what we think is that Huntington may participate in this process, and it's a scaffold. It's bringing all these components together. So you have these things you want to get rid of. They need to be put in a position where you can break them down, and that's what this autophagosome is. And then it fuses with something called a lysosome, which degrades that product. And this can then be recycled or even gotten rid of by the cell. And so Huntington may be a scaffold for this process. And so if something goes wrong with that, you have this huge buildup of material that is unwanted. And so what happens if this cellular cleaning crew goes on strike and you have this huge buildup of unwanted material? And so with that in mind as a potential function of Huntington, I just want you to keep that as we go through this talk. And this is a rather complicated slide. It's a little bit old now, but it's what I really want to do with this is just kind of point you to some of the cellular pathogenesis that might be occurring. So in red here is this expanded polyglutamine, and that you can have a normal conformation with if it's not expanded, but when you get expanded, there you get some processing of this protein that leads to these fragments that can be toxic, and that's shown by this squiggly line. And as I mentioned, maybe this is interfering with autophagy. Maybe this is causing the protein itself to dysfunction, or these toxic fragments can actually be inhibiting the autophagy. It can inhibit other cellular functions, including mitochondrial functions. It gets into the nucleus and forms these inclusions, that little orange dot in the yellow cell I showed. And then, of course, it can also disrupt the whole mechanism of genes expressing the proteins through transcriptional changes. And I also wanted to mention that when we study Huntington's disease, we're not only studying and looking at specific things going on in Huntington's disease patients, but it's applicable to many other neurodegenerative diseases, including Alzheimer's, Parkinson's, and Lou Gehrig's disease. And those all have a common factors, including this aggregation of proteins, impaired protein clearance like autophagy, loss of neurons and synapses, the place where neurons communicate with each other. There's an inflammatory component, oxidative damage, and as I mentioned, transcription changes. So anything we do that might have a benefit for Huntington's disease patients is also applicable to a number of other neurodegenerative diseases. So why even think about stem cells as a therapeutic for any neurodegenerative disorder? It's so complicated to bring them to a clinic. And one of the reasons is because so many drugs fail in our clinical trials. This is a bit of an old slide, but what I'm just kind of summarizing here is that if you're looking at Alzheimer's, Huntington's, Parkinson's, or ALS, there's clinical trial after clinical trial that just fails. We'll do the studies in mice. It looks very promising. Everybody gets excited. But then you take them to patients, and it doesn't seem to work. And so stem cells became an option for us here in California with CIRM, so California Stem Cell Agency, and I'm really proud to be a Californian because CIRM is really pushing this forward in a time when using stem cell therapies wasn't that popular. And CIRM is a $1.2 billion voted in funding to develop stem cell therapies. And if you are inclined to, I would recommend going to CIRM and learning about stem cells and how to become a champion and advocate for our research. Now, I wanted to just take a moment to bring us all to kind of the same level in thinking about stem cells. So a stem cell is a single cell that can make precise copies of itself or replicate. But it also has the ability to differentiate into mature cell types. So it can become a neuron, red blood cells, fibroblasts, all the cells that make up your body. So it has this unbelievable potential. But it's not really that simple because there's lots of different types of stem cells. And you have to think about it, really, it's the raw material to build your complete body. So there's different types of stem cells in the developing systems as well as in an adult. And here on the left side, I just wanted to talk about what a pluripotent stem cell is. So these can come from multiple avenues, including in vitro fertilized discarded eggs that were not used and implanted into a patient. And these can actually be cultured in a cell culture system as pluripotent cells, and that means they can be differentiated into any of the cell types. And so these would be considered embryonic stem cells. You can also get these cells from aborted fetuses, and those are collected from the developing brain tissue and also cultured. And I want to just take a moment here to point out this is very controversial on the use of these types of cells, and I'm going to admit that we haven't worked out all the morals and ethics surrounding the use of these cells. As a research scientist, these pluripotent stem cells from embryonic lines are available to us. The National Institute of Health has agreed upon which lines have already been propagated and utilized in research. And the more controversial is probably the fetal lines, which a lot of investigators are using, and they hold a lot of promise too. But again, the morals and ethics surrounding the use of those cells is obvious to everybody. I also want to point out that there's different types of pluripotent stem cells you can get, and many of you probably know about the induced pluripotent stem cells that you can get from skin biopsies, and many HD families and patients have been wonderful in contributing to the banks of those cells. Those cells are also potentially useful for stem cell therapies, but I think everybody probably, if they think about it for a minute, might understand why it would be difficult to get the stem cells from patients to replenish and replace into those patients, and it's because they carry the HD gene. So you'd want to either correct that, the CAG repeat, or the other option is in the future we might be able to have stem cell banks from donors, much like blood cells, that would be available for replacement therapies. And then here on the right side, I wanted to just point out the fact that there are multipotent stem cells throughout our body, including in the brain. We do have stem cells in the brain that can develop into neurons and be replenished. Heart cells have capability to be replenished. And bone marrow, of course, has been utilized for many years as life-saving treatments in replacements. And that's really a stem cell therapy. And people are also now making stem cells from fat tissue, and as I mentioned, from fibroblasts, and these methods of converting the cells back into stem celliness, if you will, into more of a pluripotent state are being explored. So the mechanism by which we really got involved with stem cells is severalfold. One is that CIRM provided the opportunities, but as I mentioned, Leslie's lab was right next to Frank LaFurla's, and Frank's lab had this wonderful postdoc named Matt Blurton-Jones who was doing some excellent work using stem cells to treat Alzheimer's disease. And Matt's idea was that he used these cells from a green fluorescent mouse. I'm not sure if you are familiar, but you can make a mouse that every single cell in its body has this green fluorescent protein. And Matt took cells from the brain of young mice and was able to develop a culturing system for those cells. And then those cells had the capacity to differentiate into neurons, astrocytes, or oligodendrocytes, the primary cells of the brain. And he could follow those cells because they were green. So he could implant these cells into Alzheimer's disease mouse models, follow them in the mouse, and see what happened to those cells. And what he showed was that those cells, the replacement cells, changed the behavior in Alzheimer's disease mice and improved their behavior. And the cells persisted and differentiated mostly into astrocytes, but also some neurons. So being next to Frank's lab and having this funding opportunity, this is when I came on with Leslie's lab. And our idea was we wanted to move away from the mouse to mouse because we wanted to develop a therapeutic that could be utilized in humans. So we started thinking about what types of human cells were available. And this diagram is really just to show you how we were initially thinking about this and what might be the potential for the stem cells. So please just let me walk you through this. So one of the first steps is you have to have a stem cell population that you can expand and grow in culture. This simple step from here to here is really very hard. And what we wanted to think about was, could we manufacture or have someone manufacture for us enough cells that could get us to a clinical trial, but also that would maintain their normalcy, if you will, because when you expand cells, sometimes they get what's called an abnormal karyotype. They get genetic modifications that could lead to tumorigenesis, which is the big, scary thing in stem cell therapies, is that you don't want to put in cells that can make tumors in your patients. So besides this ability to expand and culture, we needed cells to be able to survive an in vivo transplantation. And in this schematic, I'm really showing transplanting into multiple systems, either the spinal cord, blood, cerebral spinal fluid, but we wanted to target the brain. And then we had to think about, what would these cells do? Well, since there's such massive loss in the striatum of HD patients, we thought maybe the cells could go in and replace the lost medium spiny neurons. Another possibility was this thing called trophic support. Maybe the cells would just release factors that would support the dying cells and then keep them from dying. So that would be kind of a nursing effect, if you will. There's also, as you may know, there's this immune response in neurodegenerative diseases where there's actually inflammation that can be causing problems. Maybe our cell replacement would go in and change that immuno response and be a modulator to dampen it. And then there's neuroplasticity, which is decreased in Huntington's disease. And another possibility is maybe our stem cells would just come in and do something to increase the connections and the neuroplasticity in the diseased brain. So this was kind of our idea of thinking about this as a broad approach. And then more specifically, our first funding with CIRM was to explore different types of stem cells. So here is, I'm showing a neural stem cell, but you can push these to be more neuronal even, to make only neurons. So those are called neuroprogenitor cells. And then you can even push them to become more glial-like. And so these are glial progenitor cells. So our first project was to look at these three types of cells: neuroprogenitors, neural stem cells, and glial progenitors, and try and test the different cell types for their capability to rescue phenotypes in Huntington's disease mice. So we did all that work, and we found that the best candidate was a neural stem cell, meaning a cell that could drive to either type. And that kind of makes sense because there's lots of cell loss. Not only neurons are lost, but also glial cells. But we had difficulty in manufacturing these cells, and it just, some of the rescue didn't pan out as we expected. So this candidate looked like the best one. And so we moved forward with funding and research. And I need to mention that UC Davis manufactures these cells for us. And they do it in a good manufacturing practice laboratory, which means all the components of those growing those cells are known, and that means they're one step closer to clinical trial. And so the culturing of these cells is well worked out and done under very strict regulatory methods at UC Davis by Gerhard Bauer and Brian Fury. And the other thing I need to point out is our cells aren't naturally green. So these are not like mat cells that were fluorescing green. But because they're human cells, we can use markers for those that distinguish them in the mouse brain. So there's specific markers we can look for and find our human cells in the mouse brain so we can track what's happening to those cells. And here's our strategy. So we used Huntington's disease modeled mice, and we take these cultured cells and we use 100,000 cells directly implanted it into the striatum of these mice using a surgical procedure that's actually not that complicated. If you think, boring a hole in the skull and going down in the striatum is really challenging on the mice. They actually tolerate it very well, and we don't really see outcomes from the surgical procedure itself that are damaging to the mice. And this is also applicable even to humans because there are stereotactic devices that can be used in surgical procedures to put cells directly in the striatum. There have been other studies in other disease models where cells have been implanted directly into the brain. So it's feasible to do this in people. But then for our mice, what we do is we implant these cells and then we look at their behavior. We can collect the tissue and then we can analyze the tissue and try and figure out what's going on. And this diagram is just to show you kind of an overlay of what happens when we do the surgical procedure and put those cells directly into the striatum. We do hit the striatum very efficiently, but there are some cells in the cortex as well. This is kind of the best case image. And in some cases, we do have cells in the cortex, but that's not necessarily a bad thing. And as I mentioned, we're using a marker for the human cells here that's shown in green. But we also have other markers for things like immature neurons in red and astrocytes in blue. And our first studies in very short-term Huntington's disease mice suggested that most of our cells were becoming immature neurons, again here in red, and we're looking at the cell nucleus now in green. So you can see this beautiful human cell that is expressing this marker for immature neurons. And if you look over here, I think you can see a few cells that are blue and green, meaning they're astrocytes, but the majority of our cells look like they're becoming neurons. And this is really a short-term implant in an HD mouse model. But if we look at the long-term implant, which we've done as well, so that short-term is about 5 weeks, and this long-term now is 6 months. Amazingly, the cells are still there after 6 months. And interestingly, they're becoming postmitotic neurons, meaning they're more mature. They're maturing into neurons. And we see very few astrocytes now. And we're trying to understand what kinds of neurons these are becoming in ongoing studies. And this is just a pretty picture also showing the human nucleus and a stain that's nonspecific for a mouse cell nucleus out here. And again, these are some immature neurons. So some of the cells are immature still, even 6 months later, but they are maturing into postmitotic neurons. And this complicated slide is just to show you what happens to the mice. So up top here are graphs that are going to depict exactly what I'm going to tell you down here with these pictures. So one of the tasks we do with the mice is called a rotorod. And if you think of like the loggers that run on the log in the water and it spins, this is kind of like that, but what we're doing is we're forcing the mice to do the task because the rod increases in speed over time. And we test them for 5 minutes. A non-affected mouse can stay on this rod for the full 5 minutes. Mice that have the Huntington's disease gene will fall off pretty quickly. But if we treat the mice with neural stem cells, they stay on much longer than the untreated mice. Not as long as the wild-type mouse, but quite a bit longer, significantly longer than the untreated mice. Another task we can do is this called the pole test. So you place the mice on the top of a pole. They don't like to be out in the open and they don't like heights. So they scurry down the pole quickly. And a wild-type mouse will do this in just seconds. A Huntington's disease model mice takes longer to do this. But if we treat them with stem cells, they have a significant improvement in that task. And so these are really motor tasks that are demonstrating we're rescuing a motor impairment in these mice. Another task we can do is called grip strength. So you can kind of hold the mice out by their tail and they kind of do a Superman and they grab onto this mesh because they want to get away from you. And if you gently pull, this mesh is connected to a gauge that can measure their strength of holding on. And Huntington's disease mice have impairment in grip strength. And we can improve that with neural stem cells. And finally, I just wanted to mention this general phenotype called clasping that's just common for not only HD mice, but other mice with neurodegenerative disorders. And if you lift them by the tail, the mice with a neurodegenerative disorder will pull their hind and forelimbs into their body. Normal mice splay because, again, probably because they want to get away from you. And when we treat the mice with neural stem cells, they splay better than the untreated mice. And I don't show it here, but what I also want to mention is we collaborated with Marie-Francoise Chesley at UCLA, and she did some really beautiful studies that supported these improvements in motor functions in a long-lived mouse model. And she also found improvements in cognition. So it seems like the stem cell implants are having multiple effects, including motor and cognitive improvements. And we've now done this work in other long-term models and are trying to figure out exactly the mechanisms by which these improvements are occurring. But we do have some data in terms of what's happening to the pathology. Now remember those inclusions, this is a different stain, so the inclusions are now brown, and we're looking at inclusions in the nucleus and also in the cytosol surrounding those cells. And we found that where we implanted the neural stem cells here in the dark brown, the areas next to those implanted cells had fewer inclusions. And that was quite amazing to us. We didn't expect that finding. And so we did another type of study called a Western blot, and that's shown down here. And what I want you to think about is, remember when I talked about the scaffolding and the huge nature of the Huntington protein? Well, Joan Steffen likes to call this kind of aggregation of proteins a hairball. And she thinks that's part of the problem. You get these huge hairballs that are we can't get rid of in a Huntington's disease patient. So what I'm showing down here is the accumulation of this hairball, if you will, in protein. And that only occurs in the Huntington's disease mice. It doesn't occur in wild type mice. And what was amazing was that our treated mice had a decrease in this giant, smeary, pathological indicating hairball. And that was an unexpected outcome, really. So at this point, I just kind of wanted to stop and summarize the findings. We've seen potentially a change in these toxic fragments that are maybe impacting autophagy. We see a decrease in these inclusions. And we have some inference of a mechanism, but we don't know for sure what's going on. But there's more to this story. And that is regarding neurotrophins. So this is the nursing support that cells can do for each other. And neurotrophins promote growth at the synapse, and they're decreased in Huntington's disease. And this is an old study that just shows a factor called nerve growth factor. If you add it to a neuron, you get these beautiful branching processes. But if you don't have it in a similar neuron, you don't see that. So this could have a huge impact on synapse and communication between cells. And what we found was that our stem cells implanted into the mouse, again here in green, may be producing what's called brain-derived neurotrophic factor, one of these nursing factors. And what we don't know is that if our stem cells are stimulating the production of BDNF from other cells, or if they're actually secreting it. Now, one thing I need to tell you is BDNF is not made in the striatum. It's made in the cortex and then transported to the striatum. The cortex is the area outside of the striatum in that diagram I showed you in the first slide. So either we're stimulating the cortical cells to make BDNF, or our cells are making it and providing support directly. We don't know that yet. We also collaborated with Mike Levine and Sandra Hawley at UCLA to do electrophysiological studies. And just to summarize, you know probably that electrical currents are the way neurons communicate with each other, and they send these currents down, and then the signal goes from synapse of one neuron to the synapse of another, through the synapse of another to stimulate them. And all these complicated diagrams over here are just telling you that we can measure those electrophysiological currents. And the take-home message is that our implanted human neural stem cells have electrophysiological activity similar to immature neurons. But not only that, implantation of those cells rescue the deficits in Huntington's disease model of surrounding tissue. So the mouse cells are stimulated somehow by our neural stem cell implants to have better electrophysiology. But that's not all. All these wonderful outcomes are kind of really encouraging, but something that we did not anticipate that was amazing is shown in this slide. So we collaborated with Charlie Muschel up at Oregon Health Science University, and he did some electron microscopy for us. And I think you know that electron microscopy allows you to look at very specific details of cell interactions and cell structure. And what I'm showing here is an electron micrograph of one of our stem cells implanted here, labeled LDEND. And we know that by a specific marker. Here's another stem cell implanted. And what's amazing about this is that it's making a connection to a mouse host neural cell, and it looks like there's beautiful synapses being formed. And that in and of itself is really amazing. But this mouse cell, indicated by these purple triangles, has a marker that indicates that it's most likely coming from the cortex. So that's a mouse host cell coming all the way from the cortex, projecting into the striatum and making a connection with our human neural stem cells. And if you didn't know, the cortical striatal communication is broke down in Huntington's disease mice and patients. And if we're potentially reinvigorating that communication, it could be quite amazing. This infers that that's a possibility, but we still need more to do to verify that. So at this point, I'd just like to summarize where we are and what we're going to do with this. We're doing more long-term studies to look at the improvements and really try to understand the mechanisms that are occurring. We have to figure out the dosing. So what is the optimal dose? So we can extrapolate from a mouse to a human based on size, but that's all we can do at this point. And what we really need to know is there a minimal amount you can put in the mouse and a maximal amount? And what happens when you tweak that dosing amount above or below what we've been using as our standard? So we really need to define that. And the most important question is, are these cells safe? So we don't want to make tumors. We want to use a cell product that is safe and efficacious. So there's lots of studies that the FDA requires and that we need to do to just look at safety. So this is kind of outsourced studies in other mice that are very rigorous. And we're working with a proposal now to CERN to get the funding to do that. And then we really want to get our stem cells described as an investigational new drug, or IND, and we're working with the FDA to make that happen. So we've gone through a couple steps, a pre-IND meeting, and the FDA has given us guidance. And I want to say this is a long and challenging process, but the FDA has really been wonderful and supportive and trying to guide us in understanding what we need to do with our stem cells to get them this designation. And our funding has really come from CERN for all this work. We had an early translational award, and then we're just finishing up our preclinical award. And as I mentioned, we're applying for funds to do more studies for developing the safety work to get this to the next phase of a clinical trial. And that's called a CLIN1, and we're going to be applying for that within the next month here. And then I just wanted to point out the people that are instrumental in doing all this work. Here's Leslie, of course, our wonderful lead scientist. Here's Joan Steffen, that I've mentioned some of her work with Joseph Ochaba. You don't know me, so here I am in the background. And these are our wonderful scientists, technicians that do all the mouse work that I get to tell you about. Sylvia Young and Alice Lau are really excellent surgeons and developing all the techniques with the markers and Western analysis. And then Lexi Copen here has been culturing the cells for us when we get them from Davis. And so it's a really huge team effort, and all the members of the Thompson Lab contribute with their scientific knowledge, and we get really excited when we have outstanding results. So I'll finish there and then open it up for questions. Thank you.
Thank you so much, Dr. Riedling. That was a really fascinating talk, and it sounds like you've got a lot of very interesting evidence in mice that this could be beneficial. We're waiting for questions to come in, so please, if people have questions, ask them. We've got one from the audience here. Let's take a look. So one person wants to know whether there have been any stem cell experiments in the past on people with HD, either at UC Davis or otherwise. If you could speak a little bit to past research in humans of this type.
Yeah, so there have been actually use of fetal-derived stem cells in humans in Europe years ago, and they did show efficacy, and they actually found that the cells did survive post-mortem in several patients. So those cells, that work is very encouraging for us. The problem, it's not really a problem with it, but it demonstrates the feasibility. The issue is that we need to have methods to culture and manufacture lots of cells, and that study was somewhat limited in its scope regarding the material that you could use. So we're trying to look at methods to have a manufacturing process to make lots of stem cells for HD patients, and that's the work that's ongoing at UC Davis in collaboration with us. But there haven't been other stem cell implants in people currently, either at Davis or UCI.
Hey, Jack, this is George from HDSA. Just a quick question to follow up on your last slide, that you showed that you were having some initial discussions with the FDA to kind of prepare for a potential approach in humans, HD, for a clinical trial approach. Can you talk about what the FDA is going to be looking for to demonstrate safety for these neurostem cells prior to starting clinical trials in HD patients?
Yeah, so really you have to demonstrate that in long-term animal models you don't do any harm. And so what you do is you have to use what's called a regulatory CRC lab that's outsourced, and they're very rigorous and strictly guided by process. And they look at an implanted mouse or multiple implanted mice, and they look at all the tissues. And so they'll look at the brain and look for any pathologies or tumor development. They look at peripheral tissue and look at any effects in peripheral tissue. And essentially, if they see no evidence that the stem cells are damaging the model systems, the mice in our case, then that supports the potential of moving forward with a clinical trial. So that's the first phase, is you demonstrate in animal models that you are doing no harm. And so we're working on a proposal to do that kind of work, and it's very time consuming and costly because you have to outsource it. And so we're getting the funds to move forward with that here, and anticipate, if it all works out, that could potentially happen in a couple of years.
Thanks, Jack. So beyond mice, would the FDA, I would assume the FDA would require testing in a animal at a higher level than a mouse, like a rhesus monkey or something like that, or? So,
yeah. So interestingly, they don't require that. it can be suggested, and we think, and in our hands, we really will do that. So we plan to do some large animal studies, because even if it wasn't required, we figure we need to do our due diligence, and we don't want to move forward with a product just because we don't have to, and not test its full safety. So we plan to do some large animal studies just to really look at what happens with the surgical procedure using stem cells and how the cells move around and if there's any abnormalities. So we will do it whether the FDA requires it or not. So for us, we will, like I said, we're going to do our due diligence.
Great, thanks. We have another question about how patient donations can help with this research and whether those types of donation programs are still active. For example, if someone wanted to donate skin cells, or if they perhaps had undergone PGD IVF and were interested in donating embryos, is that possible through UCI? And how does that help with your study specifically?
I actually don't know much about the donation process. I think you at HDSA might be able to inform people better than I could about that process and what is active. I know in terms of cell biopsies and skin cell grafts or donations, Leslie is part of a consortium, and I'm sure they're still accepting donations. So what we utilize those cells, we convert them into pluripotent stem cells, and then we can use this idea of disease in a dish, where you're looking at therapeutics in cell culture that would affect different HD patients. And as you probably all know, every HD patient is a little bit different in that even if the CAG repeat length is the same from one patient to another, the onset of disease timing may be different, and the manifestation of the symptoms is different. So as many patients that could donate their fibroblasts to these consortiums would be beneficial to us, because that means we could gather more data. So I would encourage you to look into that. Unfortunately, I'm sorry I don't have the direct information of who's still working with the donations, but hope maybe HDSA can guide you on that.
So I'll just add to that we too have gotten questions about donation of embryos and cells, and there are certainly ongoing studies that are available to view in HD Trial Finder, where you can look at different mechanisms of donation of different kinds of biological samples. But embryos is one that we're also looking into to hopefully be able to provide that information. This might be a tough one, but families are certainly interested in sort of timelines of this type of work, and whether you have any sense of when we could expect potential use or trials in humans.
Yeah, so as you mentioned, that's a really hard one, and the hurdles that you have to go through for safety, although they're cumbersome, I think they're essential for stem cell therapies, because we're so in the infancy of developing this therapeutic. So we anticipate, like as I mentioned, within a year and a half to 2 years, having all the safety studies done, and then we would move. At the end of that, we would get the FDA to designate us as an IND, an investigational new drug, and with that, we would be able to move into a clinical trial of just safety, similar to what Ionis is doing, just a 1-2 safety test to make sure we do no harm. And so realistically, that could be a few years away. And then the development of the next phases to show efficacy would follow that, and that could take several years as well. But I will mention here that we are considering, and actually are going to be moving forward with testing another stem cell line that Clives Cells been using in ALS patients. He's actually in clinical trials right now, looking at safety and efficacy, and we'll be moving forward with efficacy in ALS patients. And so those cells are ahead of us, if you will. And we're going to try and see if those cells will work in Huntington's disease models. So those cells would be in the pipeline sooner than our cells. Now, early evidence says they might not be as efficacious as ours. So we still have to work this all out. It's very preliminary, but we are looking at the potential of other cell therapies, including ones that are already in trials for other diseases, to get them quickly into Huntington's disease patients. So those, if we can show efficacy and safety with those cells, we could move them within several years into an efficacy phase clinical trial.
Jack, this is George again. What are do you know the details of what Clives cells for ALS are, and how they, can you tell the audience how they differ from what you guys were using in the data that you showed today?
Yeah, so there's several differences and multiple similarities. So our cells are embryonic derived, and they're banked and cultured and made into neural stem cells. Clives cells are fetal derived, and they are also banked and cultured and differentiated into what he calls a neural precursor cell, but essentially it's very similar to our neural stem cells. And one of the differences in the literature about fetal cells versus embryonic is the fetal cells don't seem to be as efficacious and live as long and have the high potential for differentiation that embryonic stem cells have. But with that said, they still do improve impairments in models, and they look like they would be a very good candidate as a cell therapy. So those are kind of the general differences. But as I mentioned, Clives cells are much closer, and you can look them up in the clinicaltrials.gov. They're already in early phase trials.
Thanks.
Oh, sorry. Thank you.
We have a question actually about your work on autophagy that you mentioned, and someone wants to know whether your lab has experimented with drugs that support autophagy or promote autophagy to try and clean up that trash, such as the drug verapamil. Verapamil. Thank you.
We have not tried that drug, and I don't know much about it. Joan Steffen's our autophagy expert, so I could actually ask her about that. That would be really kind of the golden egg, right? If we could just change fluctuation in autophagy by drug therapies. To my knowledge, there aren't really good drugs to do that. Maybe this one that's being mentioned is one of those candidates. The really kind of difficulty working with autophagy is that there's a balance. So you need to clear enough proteins to maintain cell health, but you don't want to clear too many, right? You don't want to start to eat yourself completely. And autophagy declines with age. And so there's this balance of cleaning house versus over cleaning, if you will, that needs to be addressed. And sorry, I don't have any information about that specific drug, but I think any drugs that could alter autophagy are definitely worth exploring in our cell culture models and in our animal models.
Well, thank you so much, Dr. Riedling. We are coming to the end of our time here, and so there might be a couple more unanswered questions, but we're going to have to stop the webinar today. So I wanted to thank you very much again for sharing with us your work today, and I want to thank everyone who joined us, and this again will be available on HDSA's website and on our YouTube channel if anyone else wants to take a look at it later on. And thank you all very much. Until next time.
Thank you.
Thank you, Jack.